All right. Yeah, super excited to be here, and I want to thank the conference organizers for giving me the opportunity. I really loved the previous talk because it gave me some fond memories of my PhD work in Sally Elgin's lab, who, if y'all are familiar with her, she is credited with discovering HP1 through this, super huge, heinous cDNA screening experiment. So it was really nice to see some modeling around that. I really enjoyed that talk. Since I left her lab, I actually sort of went on to in a slightly different space in, well, I guess what we now know as quasi-silenced chromatin, so the facultative heterochromatin associated with the Polycomb machinery. And since I started my own independent research program, first at Arizona State University and now at Emory University, one of the things that I'm sort of having a lot of fun with, and the sort of different trail I decided to go down, was in sort of trying to combine principles from synthetic biology and protein engineering and bring those into the chromatin space. So that's going to be the general flavor of what I'm presenting today. So before I present our exciting new results to you, I just want to start off with a general acknowledgement of my lab. Right now we have 3 PhD students, but the work that you're going to see featured prominently comes from my student Seong Hu, or Rick Kim, who has a poster out in the lobby. So please, I encourage you all to stop by his poster and ask plenty of questions. And then I'm also going to show some work that came from efforts from some previous lab members throughout this talk. Okay, so just to start with a little bit of context, in general, the biomedical engineering challenge that we've been focusing on a lot with our work is the sort of like the general topic of epigenetic cancer therapy. So this approach to cancer therapy has gotten a lot of attention. It's gained a lot of popularity. But as I'll talk about later in my other slides, there's still some challenges to having this approach to cancer therapy reach its full potential. So just to give you a general overview, so I'm not going to explain what nucleosomes are. Thank you for all of those wonderful explanations. And I've seen a lot of beautiful cartoon illustrations of nucleosomes. These are mine, right? So that's my sort of artistic rendition. And so the general principle of epigenetic therapy, and I'm only giving you one flavor of it, right? Because you want to move, redirect gene expression in different ways depending upon the gene targets. But for this talk, we're going to focus on the approach to epigenetic therapy, where we have tumor suppressor genes that are sort of trapped transcriptionally in this in a repressed state due to the the collaboration between different chromatin modifying enzymes and the reader proteins that recognize those marks. A lot of these have actually, the ones that I have highlighted here are top targets for epigenetic therapy, and they've shown different degrees of efficacy in clinical trials. And so the general idea here is to try to release tumor suppressor encoding genes from this state and then have them converted into an active state, open chromatin, right, that's more permissive to transcription. And then so what we hope to see is through increased transcription of this family of genes, decreases in proliferation, right? So inhibition of all of the cellular phenotypes that are associated with aggressive cancer. Now, what I want to do here is sort of walk you through the mechanism of one newly FDA-approved epigenetic therapy and sort of explore a little bit, think a little bit more deeply about how it might affect different cancer cells differently. So these epigenetic inhibitors in general have shown a lot of efficacy. They've shown some really promising results in liquid cancers, but not so much in solid tumors. And then so because our lab is interested in breast cancer, we've been following the breast cancer literature very closely, looking at the impact of things like HDAC inhibitors, and then just recently these new inhibitors that are targeted against EZH2, which generates trimethylation at H3K27. So while the one EZH inhibitor is called tazemetostat, right? So it's FDA approved, looks really promising in the clinic. The clinical trials and also some of the preclinical research looking at the effect of EZH2 inhibitors on breast cancer have shown really, really disappointing results. And so I was just kind of curious about, okay, well, what can we find in the basic literature and the preclinical research that might provide some clues about why that is? Well, so these inhibitors, while they show really high specificity with the pockets that they're designed to fit into, EZH2 is not the only enzyme in the cell that has a set domain. And so you have potential for off-target effects there. There was a really interesting set of studies published a few years ago, basically asking the question, okay, well, if we allow cells to proliferate under low dose of EZH2 inhibitor, do the cells sort of gain protective mutations? Could that sort of explain a little bit of this resistance? And in these studies, they actually identified cells that carried mutations outside of the set domain that rendered the protein resistant to being inhibited by the small molecule. There's no evidence that those actually show up in people, but it's a really interesting sort of experiment, results from that. And then so finally, another thing to really think about in terms of what happens when you try to treat breast cancer cells with EZH2 inhibitors, there was a beautiful study published a few years ago where the researchers sort of asked, okay, well, what happens at the single cell level, right, as breast cancer cells are subjected to treatment with EZH2 inhibitors, specifically under hypoxia, right? That is a very specific, sort of a tumor microenvironment. And so what they saw was that, somehow the EZH2 inhibitor was binding to EZH2, and this particular drug wasn't the one designed to actually lead to its degradation, so the protein is still hanging around. But then it goes off and gains affinity for FOXM1 and then acts as a transcriptional co-activator. It's weird, right? And then the specific gene targets include matrix metalloproteases, which help to restructure the ECM, and they actually saw increased invasion out into the ECM in this particular context when cells under hypoxia were treated with EZH2 inhibitors. So that doesn't look very promising, right, for treatment. That's kind of the opposite of what you want. So all of those sort of potential challenges aside, one thing that I really think about a lot, and it's sort of more of a general, sort of a challenge with using this approach, is that, okay, let's say that you were to magically erase all of these repressors from the tumor suppressor gene that you're interested in reactivating. Well, loss of repressor proteins doesn't necessarily lead to robust expression levels in the way that would have, an impact on a cell. Maybe not in all cases. So just to walk you through that in a little bit more detail, that idea a little bit more detail, if you consider all of the things that need to happen to shift the gene from this configuration to this one, you really have to take into consideration transcriptional activators that help to create or to set up the pre-initiation complex, right? So this isn't a trivial thing. So there's a lot more to this than simply removing the repressive proteins. And this becomes even more challenging in a situation where some of these key transcriptional activators that help to set up this configuration are mutated or underexpressed, particularly in a cancer background. So one of these all look really familiar, right? P53 is mutated in half of all cancers, regardless of cancer type. And then you also have some major components of the SWI/SNF complex that are known to be mutated or misregulated in cancers. So some key components that you need to sort of install, this activated state are compromised in the cells that you're treating with epigenetic therapy. So that could be a problem, at least for some genes. Okay, so so intersynthetic biology, right? So what I'm going to, I guess, sort of talk to you about in my presentation today is how, an initial curiosity and whether or not we could actually build functional chromatin proteins sort of converge on us using this as a tool to understand what's happening in the cancer context with regulation through chromatin. So here I'm going to start with just a brief overview and also just to generally acknowledge a lot of really interesting work that had been happening, before we actually sort of started developing our application. So there is an application known as epigenome editing. A lot of you may be familiar with this. This has generated a lot of excitement. And in principle, what this approach is that you build a fusion protein where one end recognizes some DNA sequence, right? It's targeted to some gene, and then the other end carries a chromatin modifying enzyme. So in principle, you can just do sort of site-directed remodeling of chromatin, and this works, really well in certain contexts and certain applications. But I'm not going to talk about any of that today. So what I want to call your attention to is sort of this end of what I'm calling here the flow of epigenetic information, right? So you have your chromatin remodeler either adding or removing a mark in specific regions in the genome, and then so you basically sort of lay down this pattern of biochemical signals on the chromatin fiber. But here's another very important step. Now there are some cases where modification of histones in DNA appear to be sufficient to have an impact on the transcriptional state of a nearby gene. But there's a lot of evidence, I think there's probably just as much evidence showing cases where these changes don't appear to be sufficient on their own. What happens in a lot of cases is that controlling the transcriptional state, right, actually generating an output in the form of either transcriptional upregulation or downregulation is mediated, right, transduction of this signal is mediated by a family of proteins that chromatin folks call reader-effectors, right? So you'll hear them called just readers in general, but a lot of these proteins will carry one or more domains that interact with a chromatin mark, somewhere in the protein, and then in a different region of that same protein, there will be a domain that has been experimentally shown to be sufficient to cause a change and necessary, right, if you mutate it, to drive a change in expression state of the nearby gene. So we like to think of these as sort of mediators or transducers of epigenetic marks on chromatin. So what I'm going to cover today, if I have enough time, hopefully, is our work that explores this question of whether chromatin reader effectors can be rationally designed, right? So as a synthetic biologist, I'm a really big fan of this idea of sort of like isolating functional domains with certain interesting intrinsic activities, mixing and matching them, and actually building new systems that work the way that we design. So with this work, we use a single locus reporter gene model, and then we're going to transition into work that considers the an entire natural epigenome. And then so here we investigate reader effector engagement with chromatin and the response of those genes that we're hitting over time, right? So I'm going to show you some nice time course data, and we also have started looking at the sort of the spatial aspect of this using Hi-C data. Okay, so the first part. So this is where I want to kind of show you really quickly my earlier proof of concept work to ask this question of whether we can actually rationally design reader effectors based on what we know about them. So as a postdoc in Pam Silver's lab at Harvard, I started off with this protein, right? So I started with the Polycomb machinery, mostly inspired by all of the work that was coming out about, epigenetic therapy and cancer, and how chromatin was like this sort of central driver, and that got me really excited. And then also the structural data on these this particular Polycomb protein called there's CBXs, it's a whole family, CBX 2, 4, 6, 7, and 8, all share this chromodomain that have varying affinities for H3K27 trimethyl. These proteins often contain, this family contains a long disordered region that's thought to act as a scaffold for other Polycomb proteins to form the PERK1 complex. And then we heard about a chromo shadow domain. I think there's a similar domain in CBX, or maybe it behaves similarly, called a PC box. So there's a little bit of data supporting that. And so this is basically, the structure of CBX8 specifically, and this is what we started with our builds. And then all the other CBX proteins sort of share the same structure in general. So the first idea that I came up with, the first fun idea I came up with to really sort of test this idea of whether, are these things engineerable? These subdomains that were characterized, by genetic, point mutations and all like the kind of the classical approaches, and also evolutionary conservation, Well, I'm basically doing, a type of experiment that Richard Feynman, right, would have done. I don't understand it unless I can build it. So if I borrow this binding domain, clone that out, and then fuse it to an effector that has a different output, right, activation instead of repression, can I build something that binds H3K27 trimethyl enriched genes, and then instead of repressing them, turns them on, right? So this is sort of my first proof of concept. So since then, we've developed some really nice, student-friendly assays to first screen for differences in affinity for H3K27 trimethyl. And in a lysis-style assay here, we just immobilize tails at the bottom of a 96 well plate. And then we started off doing recombinant expression. This is very slow, right? And you had to purify your protein. Nowadays, we just do cell-free expression, otherwise known as TXTL, produce micro quantities and screen through dozens of these things, or at least as many as my grad student feels like screening in a week. So we've been having a lot of fun with that. And then maybe in a future talk, I can show you some really exciting results from us doing point mutations to try to find a high affinity variant of this particular domain. But for today, we're only going to be talking about the wild type version of the chromodomain, which I have here highlighted in blue. And then specifically, I'm going to show you some work where we sort of explored what happens when you allow a monovalent version of this fusion protein to interact with histone tails versus the bivalent version. And then so here we're just testing a simple idea. We're trying to basically experimentally demonstrate that bivalency of these histone binding domains, actually contribute to the affinity of the protein for the target. And then what I'm going to show you next is how these results translate into, actual transcriptional regulation. So with this in vitro assay, what we were able to demonstrate is that, you can get a, significant increase in overall, and this is sort of like approximated affinity in the bivalent versus a monovalent version. And then you also, by doing that, you don't compromise specificity, right? So the really stringent test here for specificity is H3K9 trimethyl, because the neighborhood on the histone tail looks really similar between these two. This is an AARKS motif, and over here it's TARKS, right? So very similar stretch of amino acids, but we still maintain a specificity of binding even when we double up the domain. Okay, so what does this look like in a living cell? So in order to answer that question, we borrowed a beautiful engineered cell system that was built originally by, I'm missing the name. So anyone who's familiar with this work probably know the work. I apologize to my colleague. But their lab had generated a stable cell line where a luciferase reporter was stably inserted into one site, and then in a different site, they added in a transgene that was regulated by doxycycline, sort of classical on-off switch for a cell culture, and then so that other transgene expresses a fusion protein that has a GAL4 binding domain that sits at just upstream of luciferase, and then this fusion also carries EED, the full-length coding region for EED. EED is a member of the PERK2 complex. It interacts with EZH2. And then so by targeting it to this reporter, you can generate sort of ectopically H3K27 trimethyl. This is also accompanied by epigenetic repression of expression from the luciferase gene. So the lab that generated the model showed that beautifully. We also validated it. But what we were more interested in, instead of just sort of using this as a model for Polycomb silencing, we were interested in using it as a test bed when we were sort of cycling through some early iterations of the design of this fusion protein. So here's our experimental setup. We included the monovalent, bivalent, and then also a negative control that lacked the binding domain, just to see if the cells were just if any change was an artifact of the experiment. So overall, what we showed was that, while reactivation using the monovalent version was pretty modest, it was, reproducible and statistically significant. And then we saw a pretty dramatic difference when we used the bivalent version of this fusion protein. And then so these are just some control experiments to show that, overall levels of expression between these three are the same. So the difference in luciferase activation is due to, basically the behavior of the protein and not because of differences in expression and loading. Okay, so some general take-home questions from this initial proof of concept of a synthetic reader effector, which we call synthetic reader actuators, because actuator sounds more engineering, so we like that name. So some basic, some simple but very important lessons we learned about reader effectors was that, yes, you can, this chromodomain seems to be sufficient. There was some earlier work that demonstrated that for a different fusion. And then here we sort of validated that by using it in a different context. So yes, you can streamline these. We got a functional reader effector essentially without including this sort of long extended disordered region or any of those extra features. And then we also demonstrated that the effector domain is customizable. We basically switched, a CBX8-like protein into an activator. Third lesson, which I think is a pretty important one and an interesting one, is that if you draw this out as a cartoon, right, assuming that a single nucleosome can have up to 2 H3K27 trimethyls, you would predict that the stoichiometry of activator would be higher per nucleosome versus this situation where you're occupying both targets with a single protein. And we see the opposite of what you would expect from, a naive, guess from this cartoon. And so what that tells us, maybe, with some caveats, is that really strongly suggests that stabilizing binding really does contribute in the context of a transcriptional activator specifically. And this is actually, I really like this result a lot because this sort of supports what was thought about, or some of the lessons learned from looking at natural reader effectors. More often than not, you'll see more than one histone binding domain in a single reader effector type protein. And then so this is kind of like the synthetic biology way to investigate, okay, what is the contribution of that? Why does that matter? Oh, sorry, let me go forward. Okay, and then this slide, I just want to encourage everyone to take a look at a very comprehensive review. My PhD student, Kiara Franklin, wrote recently where we wanted to, explore, okay, well, what is the potential of this, right? So we can make these sort of modular reader effectors. In what context do reader effectors really matter, right, in biomedical engineering? And so Kiara went into the literature and found all instances of cell development related disorders where a reader effector type protein was either overexpressed, underexpressed, or mutated. And this is the full list that we came up with. So this is sort of the map for the future course, right? So there's a lot of room to play and a lot of opportunities to kind of apply this platform to probing the function of chromatin through the activity of reader effectors in different contexts. Okay, so the next part of my talk, I want to walk you through some really exciting results that we published recently where we deployed our synthetic reader actuator, same configuration that I showed you before, chromodomain activator. We expressed this protein in triple negative breast cancer cells, which have been shown to respond only partially to EZH2 inhibitors. And so we were interested in sort of like treating this as, basically comparing, instead of using the inhibitor approach, taking our SRA and then using it to install robust activation at sites that had become hyper-repressed by the Polycomb machinery in the context of breast cancer. Okay, so so what I'm showing you here is transcriptome profiling of cells, BT549 triple negative breast cancer derived cells that have been transiently transfected to overexpress SRA proteins. And then so we collected total RNA 24, 48, and 72 hours after transfection. And then did NGS to identify genes that were significantly upregulated at those 3 time points. Within conditions, we get pretty good reproducibility. And then across time points, we do see some differences, right, on the outside of this Venn diagram. But for the most part, the genes that became significantly upregulated were upregulated across all 3 time points. Okay, so I was a little bit curious about these differences, right, because there's some interesting things going on there. And then we did a sort of like a time course expression profile clustering, and were able to sort of roughly identify these different groups, right? So group 0 are genes that are upregulated across 3 time points. Well, we also identified genes that were activated early and their expression fell off after the first 2 time points, ones that seem to have a delay, right, compared to the full gene set, and then a very small group where you see this increase at the middle time point, but delayed expression, and then it falls off at the last time point. So I'm going to show you that there because you're going to see a callback to this. We hadn't explored this any further in BT549, but again, we're going to revisit this idea of looking at what happens over time with a different experiment I'll share with you. But here, I'm going to share these results with you because I'm actually pretty excited about the implications that this has for, efforts to sort of improve the efficacy of epigenetic therapy for triple-negative breast cancer. What I'm showing you on this slide is just sort of a validation that the 122 genes that we saw become upregulated at any time point are actually repressed in the untreated state in the first place. So we just wanted to validate these as like real Polycomb repressed sites. So we see lower attack-seek signal in our 122 genes versus a set of highly active genes. The opposite, right, when we look at H3K27 trimethyl, and in particular in a region just upstream of the transcription start site where you would typically find like an enhancer. And then this data basically just validates that, verifies that H3K27 trimethyl at those sites depends upon EZH2, because when you treat the cells with tazemetostat, which is the EZH2 inhibitor, you see a loss of some of that H3K27 trimethyl in the same regions. All right, so then here's the $1 million question with any sort of RNA-seq type data set. What do the genes do? And then I'm going to show you some nice phenotype data so that we could answer the question of do the cells actually care, right? That you're actually pushing expression up. So our genes roughly classify into these categories that are relevant to cancer development. Most of them fall into categories that are anti-cancer, right? So cell death, apoptosis, cell cycle arrest, the inhibition of migration and invasion, and then immune surveillance. This is a really interesting one. We're thinking about ways to move this model into whole animals so that we can actually follow up with how this impacts phenotype. Now, this is a very honest figure because we also identified a group of genes that were strongly related. We looked through all the literature, and all the literature said that these are oncogenes. They are not tumor suppressors. So as expected, because our targeting is broadly distributed, right? We're not targeting based on DNA sequence, it's based on histone modification, which can show up in lots of different places. So this is not too surprising. And then we basically compare the median expression of these different groups of genes to first kind of ask the question, okay, well, these come up, but are the anti-cancer genes being upregulated more than the pro-cancer genes? And yeah, sort of, okay, but what you really want to know is, do the cells actually care, right? So what is the actual consequence of this sort of a transcriptional reprogramming? So to answer that question, we used a 3D spheroid model. We used a stable integration of a cassette that put the SRA protein under the regulation of doxycycline, right? So we could nicely, time expression and control the dose. And in this experiment, we included a negative control where we had removed the activation domain, but we left the binding domain intact so that we could tease out any effects caused by just the protein sitting at the targets. So here's just some representative examples of the data. One of the first things that we observed consistently in multiple trials was that the diameter of the spheroids that express a full-length SRA actually decrease over time. in contrast to spheroids that express this truncated, mutated version of the SRA, and they grow happily. Growth rate is the same as cells, but without adding any doxycycline at all. Forward. Okay, and then separately, we looked at invasion. So in our model, we don't do any special treatments to induce invasion. We just kind of let this happen spontaneously. So what we observed was that for all instances of any invasion events, those would show up in these negative control conditions, but for cells that were expressing full-length SRA, absolutely no invasion events, which, makes sense, right, because the spheroids are also shrinking at the same time. And this agrees nicely with some of the genes that we know have been associated with inhibiting migration and invasion. Those genes go up. Now there's this kind of weirdo gene, CCN2, that goes down. We haven't really teased out why we see downregulation of a gene in cells that are overexpressing, this activator, right? It just has an activator domain. But we do see downregulation of this gene. Maybe it's, sort of a secondary effect. And this, actually, I'm showing you this because it kind of agrees nicely with this phenotype that we observe in cells with that transcription profile. Okay, and then so to conclude, I'm going to show you some new hot off the press data that is featured in my student Rick's poster. So hopefully all of this kind of gives you a little bit more context to make the story a little bit more interesting. So we basically did the same types of experiments again, this time in another breast cancer cell line, but we picked this one to take advantage of the rich body of public omics data that's already available. Because what we're attempting to do, in order to better understand what is it about these genes that are responding to our SRA? What's their chromatin, their local chromatin environment look like? So we were really interested in looking into this because some earlier work that I didn't have time to talk about, you're probably really kind of wondering, right? Okay, if this thing behaves the way we expect, you should see it binding to all H3K27 trimethyl across the genome. And some earlier work that we published, I think the first paper was 2017, 2018, we do the Venn diagram, we don't see that. So there is some sort of selectivity happening with the SRA. It doesn't bind all regions that are identified as enriched with that mark. And then so what we've started doing recently is sort of stepping back so that we don't miss the forest for the trees. And we're interested in identifying large regions that might be more enriched for SRA upregulated genes. The reason why that is that there's a lot of really beautiful data in the literature showing that Polycomb probably has a really critical role in controlling the activity of enhancers. And you know that enhancers, right, do sort of multi-gene regulation. So we're kind of interested in seeing what kind of, picture comes into focus when we start looking at things at the broader level in 3D organized regions where there's a lot of interactions of enhancers with genes in that general neighborhood. So we're kind of like moving lower resolution than we have traditionally. First, we ask, okay, at the chromosome level, right, do you see a bias, right? Are there any sort of like chromosome level hotspots for SRAs? And then so just doing some rough math, we identified a handful of chromosomes that seem to be overrepresented or have more SRA upregulated genes than you'd expect. Oh, sorry, the red highlighted ones made us really happy because these are genes that are known classically to be targets of Polycomb, right? So your Hox genes are Polycomb regulated depending upon the differentiated cell type. So here's just a closer look at the 3 chromosomes that carry those Hox genes, and you can see when you start to zoom in an uneven distribution of SRA upregulated genes. And so this is just a snapshot of some public Hi-C data that we're starting to use to try to map these SRA upregulated genes to TADs. And then so this is some beautiful work that my student Rick had just recently done, where we're now trying to get a better handle on, okay, what are the TADs? How can we define these based on data from a CTCF binding, for instance, and also mathematical installation scores. So what really jumps out at us, so this got us really excited. So this is sort of like an expanded view of that cluster around a couple of Hox genes. There are non-Hox genes that are also activated very quickly in the early time points, all clustering together, and then you kind of peter out going this way. There is sort of mid-level H3K27 enrichment here, and then there's this really interesting region over here that is generally gene poor that has really high levels of H3K27 trimethyl. Okay, so you hear me getting excited about this. So this is just an early peek at the data. This is preliminary, and right now we're expanding our analysis out to the rest of the genome. Okay, so with that, quick plug for a, if the organizers don't mind, please offer me a couple of seconds to just kind of share this opportunity with you. I am co-chairing this year's Epigenetic Bioengineering Conference. It's in Amsterdam this year. If you're curious, feel free to ask me questions. With that, I'll acknowledge our collaborators and funders and take any questions. Oh, wait, we're not taking questions, but thank you for listening.